We're just waiting for a minute to have the people at lunch to walk over. We'll start in a minute or two. Good afternoon and welcome back to the last session for today, session 3, which is on the third main objectives that have been listed for MFCEM on regenerative medicine. We have two invited speakers, Dr. Shamik Sen and Dr. Dhirendra Rao, and we have 4 student presentations after that. So I'll start with the introduction of the first speaker, Dr. Shamik Sen. So Dr. Shamik Sen also happens to be an alumnus of IIT Kanpur. So Dr. Shamik Sen is currently a professor at the Department of Biological Sciences and Bioengineering at IIT Bombay. So he earned his BE degree from in mechanical engineering from Jadavpur University in Kolkata, and his MTech degree here at IIT Kanpur, also in mechanical engineering. Then he moved to University of Pennsylvania, or UPenn, in Philadelphia, where he earned his PhD in mechanical engineering in the lab of Professor Dennis Disher. Before joining IIT Bombay, he did his postdoctoral fellowship at UC Berkeley, which is University of California at Berkeley, in the lab of Professor Sanjay Kumar. So Professor Shamik Sen's interests or lab focuses on understanding the mechanobiology of cancer metastasis and regulation of stem cell fate, particularly how physical features of the ECM, including stiffness, porosity, topography, geometry, ligand spacing, and dimensionality influence cell behavior. With that brief introduction, I would invite Dr. Sen to come and give his presentation. Thank you.
So I would like to begin by thanking Dhirendra for inviting me to be a part of this inaugural symposium. It was a pleasure meeting so many colleagues of IIT Kanpur. So many colleagues of IIT Kanpur whom I knew before, and also many more whom I got to know here. Of course, part of lovely presentation since yesterday. And so since my master's days at IIT Kanpur, I've held IIT Kanpur very close to my heart. So whenever I get the opportunity, I do visit here. So what I'll be talking about today is our work on understanding and engineering the stem cell niche using the model system that we have worked on. So as many of you would know by now, that tissues in our body possess distinct physical properties. So if you take a tissue, just a simple extensional a measurement of how much is the strain, how much is the stress, so you can backtrack the slope of this line corresponds to the Young's modulus of the material. Of course, this is in the linear range of strain, and uh, depending on the tissues of our bodies, of course, blood is completely fluid like, brain is very soft, and as we go to stiffer tissues which are load bearing elements like bone, they are very stiff. So there is of course a very strong correlation between the physical properties of these tissues and their functions, and to a great extent this scaling in tissue, yeah, so to a great extent the scaling in tissue stiffness is very closely correlated to the collagen content which is there in a tissue. So collagen is one of the most essential elements of any tissue you talk of. So when you talk about crosstalk between the cells and the matrix, you do understand that this crosstalk is highly dynamic. To begin with, the matrix is secreted by cells themselves, right? Of course, and in the given tissue, depending on the composition and organization of the extracellular matrix, you can have distinct phenotypes. As is very evident here, this cell has aligned along the collagen fibrils and is exhibiting almost 1D like migration. The cells can also degrade or physically remodel extracellular matrix. So, this is an example where a cell is actually pulling on a single fiber. So, in a sense, this crosstalk is highly dynamic. So, during my PhD work in Dennis's lab, we had focused on understanding the role of the stiffness of this matrix. And what we showed was, if you take mesenchymal stem cells, you put them on something very soft like, which mimics the bulk stiffness of brain tissue, these cells predominantly differentiate into a neurogenic lineage. Similarly, for the stiffer tissues, they differentiate into an osteogenic lineage. In the context of cancer, which is also another area of our interest of our lab, what is very well documented now is the extreme stiffening, tenfold stiffening of the tissue surrounding the tumor. In fact, there have been seminal work done by Valerie Weaver. She was initially at UPenn, then she moved to UCSF. She showed that just by stiffening the matrix, I can actually drive cancer progression. So now this stiffness sensing, how does a cell know what is the stiffness of the tissue around it? That the cell knows by actually pulling on it. So this observation that cells sense the matrix properties was documented back in 1981 when fibroblast was seeded on thin silicone membranes, where you could see the induction of wrinkles but in a time dependent manner, suggesting that the cells are sampling their microenvironment. And this sensing is critical to stiffness differentiation of mesenchymal stem cells as well. So at one end you have the cells which is physically anchored to the outside, to the extracellular matrix, through focal adhesions. At the other end, these forces are propagated all the way to the nucleus, and at the nuclear end you have the lamina composed of lamin A and lamin B, lamin AC is the splice variant, and lamin B1, B2, which actually regulates how the chromatin within the nucleus is going to rearranged. And similar to the very strong scaling of collagen content, similarly the lamin A to B stoichiometry exhibits a scaling behavior. On very soft substrates, you have more of lamin B expression, less of lamin A expression, vice versa. So we have been focusing on this dynamic crosstalk in the context of cancer, stem cell and tissue engineering. But today I will limit my presentation to mostly whatever we have been doing in the context of embryonic stem cell differentiation. This is the work of two of my PhD students, Lakshmi Kavita Sthanam. She has graduated some time back, and my current PhD student Tanusri. So, our model system here we chose was embryonic stem cells. So, these cells are isolated at the blastocyst stage and from the inner cell mass. And so, in a sense, this is an entirely in vitro context, right? Because this whole thing is happening in vivo, and scientists have found out that if you can culture them on a layer of mouse embryonic fibroblasts, which are typically a feeder cells, then the ESCs form rounded colonies which are pluripotent, and then you can then subject them to various differentiation assays. Alternatively, these ESCs can be cultured on gelatin coated dishes, which media is supplemented with leukemia inhibitory factor. Okay, now when I started working on it, I was thinking that how is these two systems physically equivalent? Is there something which is different between these two systems? And that is where and typically the way these experiments are set up, from the time you seed your fibroblasts, it takes almost a day to 2 days for the matrix to form a confluent monolayer, and then you are seeding these ESCs. So in all probability, the ESCs are actually seeing the matrix which is secreted by the fibroblasts and not directly the fibroblasts themselves. And of course, in addition, the soluble factors which are secreted by the fibroblasts. So what we started working on was, by tuning the contractility of the MEFs, we generated what I call cell derived matrices, or MEFDMs as we call. So once the cells secrete their own matrix, we get rid of the cells, and what we are left with is our cell derived matrix. We can tune the composition or bulk properties of these MEF derived matrices, as is shown here from the ester staining or to the collagen 1 and fibronectin levels. And we then asked as to how do ESCs respond to these matrices. And what we found was, when we so leaf is plus leaf condition is a pluripotent condition. So cells are supposed to express high levels of OCT4, and in the absence of leaf, they start to differentiate. But if you see that when we supplement leaf with lysophosphatidic acid, which is an activator of rho kinase pathway, you begin to see a loss of OCT4. So suggesting that what leaf is doing is actually keeping the cells in a poised state. They are somehow preventing the cells from mechanoadapting, which by you can tweak that balance by adding LPA. So you promote cells to have more contractility, then they will begin to differentiate. So it is the poised state which is maintained by these cells. So these observations, 3D organization of the colonies which is cultured on them also show that in the presence of leaf, you have colony height is high, colony volume is more, which means that the cells are actually proliferating while maintaining their pluripotency. There was a lot more BRDU incorporation into such colonies. But on the minus leaf conditions, you see that the colony volume is less, colony height is less, which means the cells are beginning to spread laterally. And long-term culture, when we do, then we see that a stiffness dependent induction into an osteogenic lineage. So this was suggesting that the MEFDMs, first of all, the composition you can tweak, but in this particular case, we did not have much control over the fate of the ESCs that they were differentiating into. And so, but what this data showed is that you have an inverse relationship between ESC contractility and its pluripotency state. So if you can inhibit the contractility, the cells will remain in a pluripotent state. As you begin to, cells start to feel, they start to spread, and then you will have a prominent loss of pluripotency and induction of differentiation. We then next asked that, okay, can we devise a simple way in which we can change the composition and organization of the cell derived matrices itself? And we thought that why not perturb with contractile agonists which impinge on the fibroblasts, the way their shape is, the way they exert forces on the matrix. And what we generated by using various inhibitors of contractility, such as nocardazole, ML7, LPS. So, nocardazole is actually a microtubule disrupting drug, but indirectly it has been shown to activate myosin contractility. So, and what you can clearly appreciate here is, so not only is the composition of the matrices secreted by these cells distinct, but their organization is distinct as evaluated by this fiber anisotropy as well as the bulk properties. And we then did a microarray profiling of these ESCs, and what we could show was, depending on the composition of the cell derived matrices, we could actually guide them into relatively different early lineage commitment. It is not differentiation at all. This is just early lineage commitment. And uh, based on, so we actually profiled various germ layer specific markers and well as common markers, and we could show that okay, on these CFDMs there is an ecto endo meso type of differentiation. So that means that they are beginning to differentiate, but they have not exhibited any lineage specification. While in this MFDM and LFDM, which were among the stiffest, here we are seeing a prominently endodermal differentiation path. So we looked at the integrin expression of these cells, and based on the highly expressing integrins, what it seems is that most of the cells are actually early on engaging laminin and then subsequently fibronectin and collagen. So in general, when you make a 3D matrix, though we think that the ligand is there, many times it is not accessible and the cell needs to actually remodel or degrade the matrix in order to make those binding sites available. So to assay that, we looked at the expression of MMPs, and we could see again, like to our surprise, the expression of a plethora of MMPs, of which MMP11 was highestly expressed across all the MFDMs, and 2 and 17, so on and so forth. We looked at how many of these, at least among the soluble MMPs, MMP2, MMP9, which MMPs are secreted, and we could detect MMP9 in the conditioned media which the cells are secreting, which actually remodel the matrix. But to our surprise, we could not detect any MMP2, though it was present in the cell lysate. We took a closer look at the MMP2 localization, and to our surprise, we found that MMP2 primarily exhibited a nuclear localization. So even though MMPs are traditionally thought to actually be remodeling the matrix, there is increasing evidence suggesting that it is also involved in remodeling the chromatin and other functions. And these are some preliminary experiments which we did as to how does the microenvironment influence the localization of MMP, because localization will dictate its activity, what protein it is working on. And what we found that MMP2, MMP9, both of them have, first of all, on something very soft, MMP2 exhibits primarily nuclear localization. As you begin to stiffen the matrix, this MMP2 kind of exits out of the cell, as evident in the zymogram. And this localization to a great extent is actually dependent on the force on the nucleus. So if we perturb the actomyosin activity, then what we begin to see that the pore size has decreased and the nuclear retention of MMP2 has increased. So this gives a physical picture as to how MMP localization can itself dictate what it is doing. Now, when we inhibited the MMP activity, what we found was we could actually tweak the differentiation pathways. So this was the second part, where we could show that by tweaking MMP activity, we could actually redirect it from the pluripotent state, we could redirect its differentiation trajectory. Now, one intriguing observation which we had on the MFDMs was, when we culture these ESCs either on the MEFs or on the MFDMs, the genomic integrity was maintained to a much greater extent, but not so much on gelatin coated dishes, where long term culture was leading to extensive aneuploidy. And we were wondering whether this has to do with the substrate itself, because either the MEFs or the MEF derived matrices are reasonably soft in contrast to the stiff gelatin coated surfaces. So to address this question, what we did was we fabricated polyacrylamide gels, which is a well defined elasticity. We fabricated a range of them, 0.6 kilopascal, which mimics the stiffness of these ESCs themselves, and then 1 order of magnitude stiffer. So what we found was these ESCs actually mechanoadapt, so which we can either assay using the spreading. So you see on the softest surfaces, the cell spreading is nearly insignificant over a period of time, versus cells stiffen and then there's some saturation in spreading on the stiffer surfaces. And then we asked that, okay, so is there is this differential spreading driving any kind of DNA damage to the cells, and which we assessed both using gamma H2 staining as well as comet assay. And here we could see extensive DNA damage on the stiffer surfaces. What is it associated? So we first wanted to rule out that this is not because of ROS, and we use this ROS scavenger N-acetylcysteine, and we did not observe any differences in the extent of DNA damage, suggesting that there must be some other factors. And we looked at the shape of the nuclei that gave us a clue. So what you are looking at here is the side view images of the nuclei staining, and you can clearly appreciate that in contrast to the soft surfaces, as you begin to stiffen the matrix, the nucleus is extensively spread out, and which we measure here using the nuclear height. So you see a gradual drop in nuclear height. So this is a clear association suggesting that there might be a physical origin to the DNA damage. And we we assessed whether there is the compression is actually driving the DNA damage. So what we did on the very soft surfaces where the cells don't spread much, we treated them with manganese chloride, which is known to activate integrins, thereby leading to the cytoskeletal reorganization and pulling on the nucleus. So this led to a prominent drop in the nuclear height and a corresponding increase in the extent of DNA damage. Contrast, on the stiffest surface we treated with blebistatin, which relaxes the cells from exerting forces, we found the reverse effect. You see a drop in spreading area, there is increase in nuclear height and a corresponding decrease in the extent of DNA damage. So this indeed suggests that nuclear compression, which is a physical activity, is driving the DNA damage. So surprisingly, when we looked at the status of pluripotency markers, what we found was, as with increase in stiffness, at the 24-hour time point, there was a prominent drop in pluripotency markers and a prominent increase in lamin AC levels, suggesting that okay, maybe the nuclear lamella is trying to protect against this DNA damage. And this was also observed when we treated these cells with very low doses of etoposide, which induces DNA damage. So again, we found that prominent induction of lamin AC expression and prominent drop in OCT4 pluripotency markers, suggesting that this is like a mechanism which is fairly robust. Even when we did the cells, incubated the cells with known differentiating agents such as retinoic acid or aspartic acid, we asked, so do we observe such a DNA damage phenomena early on? And we did observe evidence of DNA damage early on, which then of course dropped at later time points, and this also led to induction of lamin AC expression, suggesting that this DNA damage is somehow coupled to regulation of the fate of the cells. So this is RNA-seq data which suggests that at longer time points, 70 to 72 hours, there is a differentiation into an ecto-meso lineage, though there is no stiffness specific differentiation, but there is a differentiation nonetheless. And this DNA damage, which was initially high, over a period of 72 hours, this is actually resolved. So is it that the DNA damage is initially driving a differentiation phenotype and then the DNA damage is going down? So we looked at activation of DNA repair pathways, and we found ATR was increased across all the stiffnesses over a period of time, which we verified using phosphorylation of ATR. We did not observe any activation of ATA, suggesting that DNA repair is partly driven by ATR. And what is ATR doing? So to address that question, we incubated these cells with either an ATM inhibitor Ku or an ATR inhibitor V, and what we observe is with the ATR inhibitor there is prominent drop in the lamin AC expression, suggesting that this ATR is what is regulating the lamin AC expression. So we also establish knockout lamin AC knockout ESCs, and we find that in these cases, so if you can clearly appreciate from the pictures of the nuclei, then the nuclei are very abnormally deformed, and that is, there is a corresponding increase in the extent of DNA damage. So based on these observations, we document a physical mechanism by which DNA damage induced by coupling of the cytoskeletal to the nucleus can be a driver of differentiation. And this leads, so there's a nuclear compression. This is because of the mechanoadaptation mediated nuclear cytoplasmic coupling that leads to a DNA damage response, a loss of pluripotency, laminace expression, and a subsequent activation of DNA repair factors, which then drive differentiation by induction of laminace. So, in summary, what I've discussed is showed how cell-derived matrices can not only be used as a substrate for understanding, gaining insight into how stem cell fate is regulated, but also from a very regenerative medicine application standpoint, you can use these substrates for expanding ESCs without causing genomic instability, which is an absolute requirement if you wish to use these cells for cell therapy based applications. And we are DNA damage, is a mechanism of ESC differentiation. We do see a lot of nuclear localization of MMPs. We are now trying to see whether there's any connection between MMPs and the DNA damage. So with that, I thank you. So this work I presented has been done by Kavita, an alumni, and Tanusri, and we thank DBT for funding. Thank you.
Thank you, Shamik, for a very nice talk. Floor is open for questions.
Thank you so much, Shamik. I mean, two questions for you. One is, have you looked at the chromatin topology changes using either ATAC-seq or even ChIP-seq, genome-wide ChIP-seq? So you can start looking at what are the transcription regulatory factors which are altering it. And my second question, I was very intrigued by your aneuploidy comment. I was curious to know if you looked at what is the nature of the aneuploidy in terms of the molecular composition of which chromosomal regions are replicated, and what is the consequence for the transcriptome of the cells?
Yeah, so these are very relevant questions. So to answer your first question, those experiments are currently ongoing. So we are trying to see whether these locations where this DNA damage is happening, is it stochastic or there is some specificity to the process. If it was stochastic, then that would be very dire consequences for the cell. So that we are trying to integrate. For the second part, beyond looking at the chromosome number, we have not delved deep into the aneuploidy aspects of it.
So you think it's the stiffness that is driving the aneuploidy, or is it something else that is driving the aneuploidy?
So we think that what the stiffness is doing is it is causing this DNA damage. And even though we see a differentiation, when we did a cell cycle analysis, we could see a sizable proportion of sub G1 cells. So these cells, so we need to take a closer look at these cells, suggesting that the DNA repair may be inefficient. And depending on the outcome of the DNA repair, even though it might be partly corrected, but that will lead to still survival of the cells and then ongoing of aneuploidy.
Could you use, for the stiffness part, could you use AFM to look at the alterations with the force that manifests itself subsequent downstream into your alteration in your DNA damage and other cell cycle processes?
You mean the measuring the properties of the cells and nuclei?
Yeah.
No, so which we have already done. So there is prominent cytoskeletal organization, but in embryonic stem cells, because the cells occupy the 90% of the cell volume is the nucleus.
Yeah.
So what we measure, we call it an effective stiffness.
Okay.
So we do see a prominent increase in the stiffness.
Hi, Shamik. Nice talk. So just one question. Is there a physical molecular interaction that you observe between the nucleus as well as the cytoskeleton? That's how the cytoskeleton would be imparting to the nuclear stiffness that you talk about?
So imparting the connectivity, right?
Yes.
That is by virtue of the cytoskeleton, right? We see increase in phosphorylation of myosin light chain, and that is what is causing the nuclear, it's pulling the nucleus. So the nuclear volume is roughly preserved. So as a consequence, when you're pulling on it laterally, the height will decrease.
So these physical interactions you have observed with the myosin or the actin to the nucleus?
So, in ESCs, if you do a staining, it is very diffuse. So only with perturbation experiments and these measurements, you are able to clearly say.
All right, thanks.
Hello. So hi, fantastic talk first of all. So my question was, you mentioned about the poised state, about those cells after do add LP or life, and you talk about the DNA repair pathways as how they are correlated. So do you think those cells are in a senescence mode of cells fate?
No. So when I said poised state, I meant that they are ready to differentiate, but they are not committing it. So they, in those cells, because they have not mechanoadapted at all, you are preventing the mechanoadaptation. So they are completely in pluripotent state. But whenever you remove withdrawal relief, they will start to differentiate.
Okay. So they are not in a senescence mode
No,
of.
this is a pluripotent state only.
Okay. So they and
So
also
poised to differentiate.
So you already mentioned about some subG1 stage. There, do you think they can be transformed into G0 stage so that they cannot be differentiated, and you add those differentiation marker, differentiation factor, then they start differentiating in the new forms?
That is an interesting point. We have not looked at it. We see that there is not much apoptosis at all.
Okay.
There is a steady fraction of necrotic cells. So the sub-G1 cells are actually undergoing necrosis. Okay. So we have not looked at the G0 state.
Thank you so much.
Thanks, Shamik. I'm asking from the perspective of a developmental biologist. So the way we look at it, that the cell differentiates, and along that process, it creates the ECM, which is the destination stiffness or physical characteristics for that cell type. While the data that you presented, you are providing the destination characteristics and putting embryonic stem cells in there, and you are observing that they are going towards the fate that is appropriate for that environment. So I'm a little bit curious to know that why should the cells do that, because normally the cells would be coaxed by other signaling molecules, and they will secrete the ECM, create the physical stiffness, and go further. So just I thought that I'd like to know from
I
you.
think this is a very valid concern. So from the perspective of usage of these stiffness controlled substrates, the reason why they were used was you could actually control their stiffness and hold it. Okay, but that said, that one of the critical aspects of the in vivo environment which they don't recapitulate is the remodeling. Okay, and that is why the cell derived matrices serves as a good source for us to assay, because there is an existing matrix, but this is also constantly remodeled by the ESCs. So a data that I did not show you is that in the context of ESCs on these different cell derived matrices, within a period of 4 days, whatever was the initial stiffness of the matrix, it dropped nearly by 50 to 75%. So ESCs are actively remodeling that. That is an aspect which is missing in the PHL studies. But the reason we had to follow this approach, because when you are doing with cell derived matrices for which we can vary the stiffness, there's also so much of composition which is variable. Lot of growth factors are actually sequestered within the matrix, which makes it very difficult to parse the role only of stiffness. That's the reason why we kind of use these in vitro substrates to gain some insight. But then even on the cell derived matrices, when we went and looked at the gamma H2X level, there also we see that there is this gamma H2X induction, suggesting that this DNA damage seems to be an early program. So the way I would like to think of it, is it that the DNA damage is enabling reorganizing of the chromatin, which is so that then you have genes which are existing in multiple chromosomes can be brought closer and co-regulated. So, so the next step is to actually identify what is there some rule as to which of these sites in the chromatin are particularly susceptible to forces. So that way they can dictate the way this fate will be regulated.
Yeah, so thanks, Shamik. Nice work and very great observation. So my question is related to the way experiments are done. So did you first coat the surface and then you grew the cells on top of that, or you first grew the cell and then you put the material to study the?
In which context are you asking?
So wherever you are measuring this mechanical.
No, so of course we always coat the surface. See,
Right.
these PA gels are inert. You have to functionalize it with your ligand of choice.
Right. So if you are doing that way, don't you think that testing should be when it is in the middle of the same material rather than being on the top of the surface?
You mean in a completely 3D context?
Yeah, right. So then probably it might give some other kind of
so
interaction.
of course, so the dimensionality angle will matter. So in that context, the cell-derived matrix is a much, it really recapitulates the in vivo environment in terms of its molecular and organizational complexity.
Yeah, so I'm coming from the context that because if you are growing on top of the material, then it creates a interface of the fluid and the semi-solid, and then their cells are growing, and then
no, so the cells are in physical contact with the substrate. The fluid, you mean the media itself?
Yeah, on top of it you will having the
yeah, of course the cells are submerged.
Right. So if they are having mechanical interaction both sides on the top of the product.
For that one needs to do a complete 3D culture.
Right.
Okay.
So
but the problem, main challenge with 3D culture is whenever you want to subject it to any kind of molecular process, just getting the RNA yield will be very low, so you will be completely handicapped.
Thank you. Thanks.
So our next speaker is our very own Dr. Jayendran Rao. So quickly to introduce Dr. Rao to you. So Dr. Rao is currently a professor